ସାର୍ ମାଇକ୍ ଅଫ୍ ଅଛି, ଶୁଣାଯାଉନି। ସାର୍ ଆପଣଙ୍କ ମାଇକ୍ ଅଫ୍ କରୁଛନ୍ତି। ସାର୍ ଶୁଣାଯାଉନି କିଛି।
Okay, right.
Right ଅଛି
ହେଲା ନା?
ନା ଅଫ୍ ଅଛି ଏଇଟା ଅଫ୍ ଅଛି
ଏଇଟା ତ ଅଛି ରାଇଟ୍। ଦେଖୁଛ ମୋର? ୟେସ୍, ହେଲା ଆସିବ ମୋର ଏଇଟା।
କ୍ଲାସରୁମ୍
କ୍ଲାସରୁମ୍ ଟା ଅଫ୍ ଅଛି।
ହଁ ହଁ ଏଇଟା ବଜାରରେ ଠିକ୍ ଅଛି
So am I audible? Hello?
Yes, sir. Yes, sir.
Okay, right. Okay. Yes, right. Sorry. There is a major network problem today. That's why it should delay. Okay. I'll try my level best to complete this block today. Today's block is that is what? About plant taxonomy, which includes tools and evidences. Tools means what? How a taxonomist use the process of what collection, etc. And that is first, second time that is topic is what? Tools and evidences. Mark it. Block 3. Field observation and ecological and phytosociological. Okay. Under the categories tools and evidences, first that is that is herbaria and museums. Mark the word herbaria and museums. What is a herbarium? A herbarium is a collection of pressed and dried plants arranged according to some valid system of classification and available for reference. Again, that I am telling that herbarium and museum. A herbarium is a collection of pressed and dried plants arranged according to some valid system of classification and available for reference. All research and educational institutions related to plant species have their own specimen collected locally and also billions of gradually accumulated specimens which documents the flora of one or more continents. That means totally actually herbarium is what? That is total collection of flora. Collection of flora means first you take some samples, then dried it, then press it, then keep it inside the what? Laboratory in a systematic way. That is called what? The collection of that is called herbarium. And herbaria began early in the 16th century in Italy as collection of dried plants. Okay. Linnaeus, who is known as the father of taxonomy, popularized the current practice of mounting specimen on a single sheet of paper and storing them in stock. Okay. Herbaria are the permanent repository of plant specimen and a source of information about plant and vegetation. What is the purpose of the herbaria? Herbaria are the permanent repository of plant specimens and a source of information about plant and vegetation. Okay, in the following subsections, we will discuss the methodology you should adopt while going on the field trip for collection of plants and animal specimens. Also, you will be given some information of National Botanical Research Institute, which is located where? In the Lucknow. The second point of that is tool, taxonomy tool, that is what? Field methodology. Field methodology, second point. First point that is called what? Herbaria and museum. The second one is field methodology. Before you plan for a field trip, decide the tools and equipment required. Certain equipments are necessary for plant collection. What are the equipments required for the plant collection? That is, a collecting pick is required for the digging of root and rhizomes. Collecting pick, which is responsible for the digging of the rhizomes. Rhizome what? Modified stem. Okay. A strong knife is required for cutting branches and other plant parts. A pair of pruning shears is needed for cutting woody and hard materials. A pair of forceps is needed for opening the floorboard to study the stamens and carpels. A basculum is needed for accommodating collected specimen to be studied and pressed. A plant pressed together with blotters and newsprints for pressing and keeping the collected specimens. Also, you should carry with you a field of book for noting down the details of collected specimens. Polythene bags in good numbers are also needed for keeping fresh plant specimens. After storing plants in these bags, the mouth of bags should be tightly closed to avoid wilting. For collecting aquatics from ponds and lakes, carry a long rope, and for collecting cones, fruits, and fungi specimen, carry bottles. Obtain necessary permission from the authorities concerned to visit a forest. Ask for forest guide or any other official assistance. Okay. Always keep a copy of all essential documents in your camp. Make proper arrangements for your stay, food, clothing, and work field. Because topic is market. So today's topic is tools, to taxonomic tool. That's why it may be not interesting, but you have to go through it. Okay. Again, best period of collection of plants from February to September. Mark this. Collection of plants that is from February to September. You should know that our motherland is rich in flora and fauna. Look for plants in rocky crevices, sand dunes, marshy regions, mountains, and even on dead and fallen trunks. Observe the nature of the plant, its association with other plants, and subtract. Collect complete and perfect material with flowers and fruits. While collecting medicinally valuable plants, collect sufficient quantities of roots, shoot, seed, and plant parts, which is of medicinal value. Later on, you can sow the seeds in the gardens and raise plants to add your collections. If the size of the plant exceeds the size of the mounting board or herbarium sheet, bend it in N or U form, but don't break the stem. Collect phytoplanktons by slowly towing fine net. Okay. A small funnel container at the tip collect organisms. Now preserve the catch in iodine, potassium iodide, glacial acetic acid, formalin, and water. Collect seaweeds in polythene bags, properly numbered, and preserve these in 2.5% formalin alcohol. Collect fungi specimen as a whole, fresh and healthy along with the substratum or host. While collecting lichens, note the name of the plant serving as the substrate. Preserve liverworts and mosses by drying and cactus and other succulent in liquid preservatives. Okay. If you collect small fleshy fruits, pick them at several places and dip in hot water till whole juice exudes out. Now you press these specimen, wrap aquatic plants in damp newspapers and keep them in basculum until they are pressed. Okay. So these are the how you collect the plant and how you preserve it. Okay. For collecting insect, carry bottles and some killing agents like ethylene acetate and chloroform with you and kill the insect immediately without affecting their color. Mark it. Label the bottles as poison because all these killing agents are deadly poisonous. Don't leave insect in the bottle for longer time, and there should be no overcrowding of specimen in the killing bottles. Okay, that's about how to collect the specimen and how to preserve it. The second point, that is what? National Botanical Research Institute, Lucknow. Mark, the topic is marked. Today's topic is tools and evidences. Okay, that's National Botanical Research Institute, Lucknow, which was earlier known as National Botanical Gardens. The previous name of Botanical Research Institute, Lucknow, that is known as Botanical Gardens. And prior to this name, the garden was known as Sikandar Bagh. Sikandar Bagh. And Professor Ken Cole was the founder director of this garden. That's an important point. Not to be again, I am repeating that is an objective question, full of objective question. This one, National Botanical Research Institute, Lucknow. And previously, it was known as National Botanical Gardens. Prior to this name, it was known as Sikandar Bagh. And Professor K.N. Cole was the founder director of this garden. The general plan and layout of the garden is sufficient proof of his aesthetic taste and creativity. Dr. T.N. Khushbu took over as the second director of the garden. Where the garden situated? The garden situated on the banks of the Gomati in the heart of the city of Lucknow. It grows and preserves a rich collection of diverse plants. These specialities of the gardens are palm. What is the importance for this of this garden? What are the richest source of this garden? These are palm, ferns, medicinal plants, orchids, cactus, and other ornamentals. Okay. Now the garden is funded by Central CSIR. The garden is funded by CSIR, Central Scientific Research, Central Scientific and Industrial Research. It is a temple of learning and research in India. Okay, that is. The next, that is what? The first, that is National Botanical Garden Research Institute. Then what? Second point, gardens and greenhouse. Gardens and what? Greenhouse. Public parks and gardens are center of aesthetic value or aesthetic beauty and serves as a place of recreation. Botanic garden is a living repository of plants and arranged and maintained on scientific basis with accurate taxonomic identification of plants. Okay. A good botanic garden explains, what is the explanation of the good botanic gardens? First, number one, importance of afforestation, evolution of cultivation of plants, horticultural practice for a layman, housewives to get interest in gardening, etc. Okay. Then what is the greenhouse? Greenhouse is a place to grow plants in desired environment. Again, I'm telling that. I'm telling the greenhouse effect. Greenhouse. Greenhouse is a place to grow plants in desired environment. In temperate countries, plenty of air and light is provided to the plants, but in tropical countries, it is meant for shade-loving plants. The essential elements of a successful greenhouse environment are heat, humidity, ventilation, light, and managed in the method of arranging plants. The heating, humidifying, watering, and introduction of the nutrients are all done automatically. Greenhouse soil is lighter and better aerated, richer in nutrients than the garden soil, and well made to compost sand, etc. Okay, that's about the greenhouse. Then Botanic Garden Q, that is London. Next, Botanic Garden Shivpur, that is India. And actually, what is the role of botanic gardens? Role of botanic gardens. The first one, what is the role of the botanic gardens? First, number one, cultivate and maintain plants of interest on scientific basis. Second, show vegetation of the world on geographical characteristics, that is Himalayan flora, alpine flora, etc. Third, maintain plants which are in danger of extinction and save species. Fourth, plan germplasm collection. Next, serve as research centers in various fields. Facilitate taxonomic studies by preserved and live plants. Promote educational program and research in experimental botany and ornamental horticulture. Exchange of materials, live, dried, and index-based sediments, are a list of seed offered for exchange. Okay, that's about. That about the botanical gardens. Then about the zoological parks, because it is become to life science, that's why your zoological parks. Then under the zoological park, it comes that is your wildlife sanctuaries. Wildlife sanctuaries. In this category, you have to remember number of wildlife categories. Your Kaziranga Wildlife Sanctuaries, Manas Wildlife Sanctuaries, which is famous for tiger reserves, Jaldapura Wildlife Sanctuaries, okay, Point Kalimala Wildlife Sanctuaries, Periyar Wildlife Sanctuaries, okay, Bandipur Wildlife Sanctuaries, and Sesam Gir, okay. Then Anamalai Sanctuaries, that is what wildlife sanctuaries. Then come to what? That is national parks. So a question may be asked, what is the difference between the sanctuaries and national parks? What is national park? National parks is an area dedicated to conserve environment, natural and historical objects, and to conserve the wildlife then. That is national park. National park also provide for the enjoyment from animals in such a manner, and by such means as we leave them unimpaired for the enjoyment of future generation. National park is the best example of what? That is in situ conservation. In situ conservation. That is, animals are allowed to grow under its natural environment. Okay. And what are some important national parks of India? It may be Hazaribagh National Park, Similipal National Park. Now, of course, it is Similipal National Park. Now it is known as Similipal Biosphere Reserve. But previously it was Similipal National Park. Then Palamu National Park, Kanha National Park, Tendova National Park, Bharatpur Bird Sanctuaries, and Corbett National Park, which is the famous wildlife sanctuaries and was constituted in the 1935. And the first national park of India, that is what? Corbett National Park. So next point, collection, leveling, mounting, identification, and care of taxonomic collection. Because mark, the topic is tools and evidences of tools and evidences. These are the tools. That is next one, collection, leveling, mounting, identification, and care of care of taxonomic collections. Okay. Collection of plant specimen is essential for taxonomic research. Herbarium specimen become a permanent record. Again, I am telling that. Herbarium specimen become a permanent record. Select the plant material carefully. The specimen collected should always be in the flowering and fruiting stage. At least 5 specimens of each element should be collected, preferably in different stages of flowering and fruiting. Preserve the specimen by pressing it as soon as it is collected from the site. Observe the pattern produced by hybridization, changes in the soil moisture, slope, and light. For collecting specimens in their different stages of flowering and fruiting, it is necessary to visit the locality several times during a year. The leaves, flower, fruits, and seeds of flowering plants are important. These will make the identification of specimen easy. Similarly, collect insects flying towards a torchlight and in the evening by using ultraviolet lamps. Also use mist nets for collecting the birds. Okay. While collecting the specimen, use good judgment and don't collect rare or uncommon. Mark the word. Don't collect rare or uncommon plants. You should know that some species have become extinct at a particular site because of thoughtless collections. Then what happened after the collection of the material? Now, press the specimen either by using plant press that you have to purchase from the biological supply house, or you construct it out on a plywood sheet or cut into 12 by 18 inches pieces to be used for either end of the press. That means second point is what? Pressing. Okay, press the plants. Then you can also protect your specimen in the herbarium cases by providing adequate heating arrangements. It is not possible to mount all plant materials directly on herbarium sheets. Preserve them in wooden boxes. Preserve dry fruits in envelopes and label them. Preserve succulent materials in liquid preservative made of 5% aqueous solution of the commercial formaldehyde. Okay. Arrange plants in the herbarium according to the selected classification. Keep all the specimens, both plants and animals, that you have collected from one locality together. Okay. The next is what? Type specimen. Next point, type specimen. The specimen on which, mark the word, the specimen on which the names of the species are based are kept as type specimen. Don't replace the specimen because these will serve as reference materials for the species identified. Store the specimen in safe and standard museums, apparently, from other general collection, and handle them with great care. That is type specimen. Okay. The next, that is what? Tools of a taxonomist. Second part. Tools of a taxonomist, second part. That is what? Introduction, that is first. Tools of a taxonomy. Second part, that is laboratory oriented. Laboratory oriented. Under this category, you have to go through morphological evidence. The topic is marked here. Again, I'm telling that today's topic is plant taxonomy, tools and evidences. Very complicated topic, and it is having no interest topic, not having no such interest, that is topic is not actually enjoyable, but you have to go through it in detail. Tools of a taxonomist, that means topic is tools and evidences. So, tools of a taxonomist, second part, this one, that is laboratory oriented. Laboratory oriented means that is, it comes under morphological evidence, anatomical evidence, mark the word, morphological evidence, anatomical evidence, palynological evidence, embryological evidence, cytological evidence, paleobotanical evidence, physiological evidence, ecological evidence. Okay, that is the tools of a taxonomist, laboratory oriented. Again, I am telling you, laboratory oriented, morphological evidence, anatomical evidence, palynological evidence, embryological evidence, cytological evidence, paleobotanical evidence, physiological evidence, ecological evidence. That is the second point, that is taxonomic library documentation, reference work in plant taxonomy. That is general taxonomical index, first point. Floras, monographs, revisions, manuals, periodicals, dictionaries, reference work in animal taxonomy, international truth of zoological nomenclature, guide to the generals. The next point, that is keys to identification. After collecting it, how will you identify it? Key to identify the plants and animals. What are the keys? Types of keys. We'll go through it about this. And construction of keys, how to use a key, how to prepare a key, and punch cards. Okay. Then last one, herbarium ethics. That is collection of specimen, borrowed specimen, exchange of materials, relationship with what? Co-workers. Okay. So let us go simple one by one. But first, that is what? The laboratory oriented. First, that is rules of attack, that is laboratory oriented. First, morphological evidence. Morphological evidence, that is okay. In the morphological evidence, that is what? It is the study of structure of plants and animals, usually dealing with the organism and its components organs. Morphological evidence provides the basic language for plant characterizations, identification, classification, and relationships. That is morphological evidences. Okay. Then second point, that is your what? Anatomical evidence. Anatomical evidence, that is anatomy is the study of the structure. You must have gone through the anatomy of stem, leaf, root, etc. That is the anatomical evidence. That is means what? But it is also having some important features. What is? In monocots, you know that anatomical evidence. In monocots, and what? The vascular bundles are closed and scattered. In dicots, the vascular bundles are conjoint, collateral, and open. Okay. In some dicots, vascular cambium will form between the bundles, eventually connecting throughout, forming the solid ring conducting the tissues, bicollateral vascular bundle. The presence and structure of trichomes as well as their distribution patterns among taxa are taxonomically very important. Mark the presence of trichomes. That means the presence of hair on the stem is one of the important characteristics. Okay. Paleontological evidence. Paleontological evidence means what? Study of pollen grain, that is paleontology. Paleontology is the study of pollen and spores. The taxonomic characters of pollen grains includes wall structure, polarity, symmetry, shape, and grain size. In angiosperms, pollen grains are of two kinds, that is monocouplet and tricouplet. Monocouplet pollen grains are boat-shaped, have one long germinal furrow and one germinal aperture. This is characteristic of primitive dicotyledons and majority of the monocotyledons. According to some paleontologists, the first flowering plants had monocouplet pollen grains, tricouplet grains, and globules. Okay. देखो ମୁଁ खाली basically point गुड़ाक discussion कर दूची. You go through your study material, that is. Okay. That is about anatomical evidence. In case of embryological evidence, embryology is the study of what? Successive stages of sporogenesis, gametogenesis, and the growth and development of embryo. Okay, the number of nuclei in the pollen grains at the time of pollen dispersal is either 2 or 3. Double fertilization is an example of embryological unity throughout the angiosperms. Okay. Then cytological evidence. Cytology is the study of morphology and physiological of the cells. The information about the chromosome number, shape, pair, and meiosis is used for the classification purpose. Okay. Then paleobotanical evidence. Paleobotany is the study of plant fossils. Study of plant fossils. Okay. The best example that is study of plant fossils. That is paleobotany. And paleobotany uses microfossils such as pollen or macrofossils of leaves, stems, and other plant parts as a source of data. Okay. That is about paleobotanical evidence. Then physiological evidence. Physiological evidence and biochemical evidence is providing data of increasing importance to plant systematics. Recently, it has become apparent that a syndrome of anatomical and physiological features related to A high efficiency carbon fixation process occurs in large number of plants. Okay, that is physiological evidence. Then your last point, that is what? Ecological evidence. Again, I'm telling that today's topic is tools and evidences. Tools and evidences. Okay, that is. Next, what? Ecological evidences. Ecology is concerned with organisms in relation to their, last class I already have told that the study of living organisms with relation to the environment. Now, what are the important role for the ecology with relation to your taxonomies? That is, ecological studies demonstrate that the character states of many morphological features are correlated with environmental factors such as light, moisture, and soil fertility. The distribution of taxa, both plants and animals, the variation within the taxa and adaptations. Okay. In animals, every species has its nearest relatives in food preference, breeding seasons, tolerance to various physical factors, etc. Okay, that is about ecological evidence. All evidence are above. The next document, what? Taxonomy library documentation. Taxonomy is fundamentally a descriptive and highly documented science. For this reason, its literature is voluminous and constitutes a vital part of its structure. There is no complete nor even adequate bibliography dealing with exclusively with the taxonomy literature. Okay. What is the general taxonomic index? General taxonomic index. The index serves as an aid to locate quickly the source of original publication of a name, to learn if a particular name has been applied to a plant or to which order, family, subfamily, or tribe plant of a given name may belong. Important index of the vascular plants, that is, okay. Then what? Monographs. What is monographs? A monograph is defined as the complete accounts as can be made at a given time of any one family, tribe, or genus, nothing being neglected of it. That is a monograph. Okay. Then periodicals, then manuals. That is not necessary. Okay. So next point, that is what? Keys for identification. How you will identify the plants. Keys are determined as a device to help in identifying plants and animals. These are chiefly an aid to identification based on successive choice between two contrasting characters. In plant, keys are a part of floras or monographs given before the description of taxa and comprising statements in copulate which serve as a lead. Okay. Suppose now I for key 1. What is key 1? Plant with net band leaves, floral leaves in 4 or 5, embryos with 2 or more cotyledons, that is dicot. Number 2, parallel or reticular venations. If parallel, that is monocot. If reticular, that is dicot. Number key 1. Perianth, which is not perianth, there is no distinction between what? Calyx and corolla. Then whether petals are fused or what, apopetalous or gammapetalous, that is the key 1. And in case of key 2, what is? Plants with band leaves, floral leaves in 4 or 5, embryos with 2 or more, the same pattern, that is. But plants, okay, that is key 2. The key 3, wings mostly haline, that is, okay. Wings mostly opaque, etc. That is. They go through in the study material, that is because it is such a type of that is syllabus, that is only line by line you have to go through the study material, or less quite impossible to explain in detail, that is. Then the construction of the key. How to use a key? The use of a key is analogous to traveling a highway that forks repeatedly, each fork having roadside directions. If a traveler follows the proper direction, the destination will be reached. Okay. The first step in identification of an unknown plant involves the use of keys to determine the family. Next is the key to generate which will provide the generic name. After the genus is determined, the keying process is repeated within the genus for determination of the species. Okay. Then how to prepare a key? Let us prepare a key by making a choice of characters, listing significant and insignificant characters, tabulating these characters and forming dichotomies. Okay. That is. Then your punch card. Punch card means what? Punch cards are used with the number of text to be keyed out is large. In this type of card, the holes at number corresponding to a character are made open by cutting the perforation into the margins. Then herbarium ethics. Okay. Then collection of specimen. Collection of plant specimen is essentially for taxonomic research. Okay. These circumscribed species and documents their viability. Plant materials is carefully selected, pressed, and preserved. While collecting the specimen, both plants and animals, locations, and habitats information is recorded. Some features like flower colors and fragrance is noted because these are evident in living plants and are not easily observed in dried materials. Okay, that's about. Then borrowed materials, exchange of materials, relationship with co-workers. All these are come from which category? That is tools of a taxonomist. Okay. That is actually not interesting topic at all, but totally data based. You cannot teach it by your own accord. मैं निजो इच्छा रे तुम पढ़े परिबो ना जहाँ अच्छी मटेरियल केवल सेटा लाइन बाय लाइन जहाँ करियो बिकॉज़ इट इज द टॉपिक टूल्स एंड एविडेंसेस खाली जस्ट ओवरऑल आईडिया ता मु देई देली if you are having study material you can go through the in detail. Okay, and tomorrow class perhaps detail about the taxonomy last part. Satyada যদি কিছু doubt থো, if you are having any doubt again you can ask me. Hello, Honda Hare Krishna. Hello.
Yes, sir.
Hello, Hare Krishna.
Yes, sir.
Am I audible? ଶୁଭା ଯାଉଛି ତ?
ହଁ sir.
ଆଉ କଣଟା କିଛି ନାହିଁ ଡାଉଟ୍ ନାହିଁ କିଛି କହ. Hello, Nurjan. नहीं. Okay. Right. पूरा थरो स्टडी हो जाओ पूरा पढ़ी सारा परे लाइन बाय लाइन काने चैप्टर एक्चुअली बोरिंग चैप्टर टा टैक्सोनॉमी टा जेमटा विशेष करी एई चैप्टर टूल्स एंड एविडेंसेस टोटली बोरिंग चैप्टर बट यू हैव टू गो थ्रू इट. इट इज आल्सो इट इज टोटली फुल ऑफ योर ऑब्जेक्टिव क्वेश्चंस. लॉन्ग क्वेश्चन टा वेरी शॉर्टली रेयरली बट फुल ऑफ ऑब्जेक्टिव क्वेश्चन दैट इज दिस चैप्टर. ओके, एनीटा let us wind up. Again we will meet tomorrow in the last class. Okay, thank you.
Thank you, sir.